Hi, good evening. Welcome back to CSIR-NET preparations. I'm Dr. Bobby on behalf of New Age Cosmetic White EdTech Healthcare Consultancy. I welcome all of you and to attend my daily lecture notes. Today we are dealing with Unit 13, which is titled as the methods in biology. The topics covered under this title are 1. Molecular biology and recombinant DNA methods, histochemical and immunotechniques, biophysical methods, statistical methods, radiolabeling techniques, microscopic techniques, electrophysiological methods, methods in field biology, and computational methods. Okay, going over and starting out with molecular biology and recombinant DNA methods. Well, the syllabus of molecular biology and recombinant DNA methods would be including the following details: syllabus, isolation and purification of nucleic acid, RNA and DNA, analysis of RNA, DNA and proteins, gel electrophoresis, isoelectric focusing, molecular cloning, and preparing cDNA libraries, plasmid vectors, PAGMIDs or PAGMIDs, COSMID and YACs, expression of recombinant proteins, isolation of specific nucleic acid sequences, detection of post-translation modification of proteins, DNA sequencing, site-directed in vitro mutagenesis, gene knockout microarray, isolation, separation, analysis of carbohydrates and lipids. DNA technology is the chemical manipulation of the genotypes and resulting phenotypes of organisms such that living organisms are modified. Alternatively, no longer living organisms or their no longer living parts may be analyzed chemically at the level of genotype. Right, and the use of DNA technology has revolutionized how scientists study the genetics, biochemistry, even the ecology and evolutionary biology of organisms, plus has allowed the development of novel biological products. Indeed, whole industries are now devoted to DNA technology based on production and analysis of biological materials. Together, the manipulation and analysis associated with DNA technology, genetic engineering, and biotechnology are based on a number of technologies generally referred to as molecular techniques. Molecular techniques include, though are not limited to, 1. Gene cloning, which is associated with various molecular techniques, including in vitro restriction, enzyme digests, and DNA ligation, plus additional, less artificial manipulations, including transformation and transduction. Creation of DNA, polymerase chain reaction, gel electrophoresis, various blotting techniques, southern blotting, northern blotting, western blotting. RFLP analysis, DNA sequencing, isolation and purification of nucleic acids, DNA and RNA. 3 major types of techniques or combinations of them are employed in the isolation of nucleic acids: differential solubility, absorption methods, or density gradient centrifugation. The choice of method would depend on the type of DNA being isolated and the application. A major goal of nucleic acid isolation is the removal of proteins. The separation of nucleic acids from proteins is generally easily accomplished due to their different chemical properties. In particular, the highly charged phosphate backbone makes the nucleic acids rather hydrophilic as compared to proteins, which are more hydrophobic. Separating the different types of nucleic acids can be more problematic in that they all have similar chemistries. On the other hand, though this similar chemistry results in a few basic procedures which are common to many nucleic acid isolation protocols. Most nucleic acid isolation protocols involve a cell lysis step, enzymatic treatments, differential solubility, example phenol extraction or absorption to a solid support and precipitation. Cell lysis. Nucleic acids must be solubilized from cells or other biological material. This solubilization is usually carried out under denaturing conditions such as SDS, alkali, boiling, or choreotropic agents. These denaturing conditions efficiently solubilize the nucleic acids and generally do not adversely affect them. In addition, the denaturing conditions promote the removal of proteins during the subsequent steps and inhibit the activity of nucleases which would degrade the nucleic acids. Enzymatic treatment. Another approach is the isolation of nucleic acids. Another approach in the isolation of nucleic acids is to degrade unwanted components. For example, inclusion of proteases, example proteinase K in the lysate will promote the removal of proteins. Proteinase K is still active at 55 degrees centigrade in the presence of 0.5% SDS. The elevated temperature and SDS improve solubility and inhibit any DNA activity that may be present in the lysate. Nucleases can also be used to remove unwanted nucleic acids. For example, many DNA extraction protocols include a RNase treatment step and vice versa. It is important that the RNase be free of DNase activity. DNase-free RNase is easily prepared by boiling commercial RNase for 10 minutes. The stability of RNase makes the preparation of RNase-free DNA more difficult. RNase-free DNA should be purchased from a reliable vendor or tested before it is used. Phenol extraction. Phenol is an organic solvent that is used to separate proteins from nucleic acids. Proteins are hydrophobic and partition in the organic phase. Nucleic acids are highly charged and partition in the aqueous phase. The advantages of phenol extraction are that it is easy to carry out and can be adapted to many applications. It is also easily applied over a wide range of volumes, 40 microliter to several milliliters. And in particular, phenol extraction is widely used for the isolation of high molecular weight genomic DNA. Phenol extraction is accomplished by mixing the sample with an acre volume of phenol, which has been previously saturated, with a twist buffer at pH 8 containing EDTA and sodium chloride. The phenol should be molecular biology grade phenols, and store at minus 20 degrees centigrade until preparing the saturated solutions. The saturated solution is stored at 4 degrees centigrade. Phenol is easily oxidized as evidenced by yellowing, and the oxidation product can break DNA. Oxidized phenol should be discarded depending on the application. The two phases are completely mixed by vortexing or gently mixed. Example, high molecular weight DNA. The phases are separated by centrifugation, and the upper aqueous phase, which contains the nucleic acid, is retained. Proteins will often be visible as flocculent material at the top of the phenol phase. The two phases need to be carefully separated in that the nucleic acids and proteins tend to be at the interface. Leaving too much of the aqueous layer behind will lead to an undue loss of material, and aspirating too close to the interface can include proteins. The aqueous phase can be re-extracted with the phenol to remove more protein. Phenol is a hazardous waste material that needs to be disposed of properly. A common variation of phenyl extraction is a mixture of phenol chloroform, isoamyl alcohol 25:24:1. The more organic chloroform removes lipids, denatures more protein, and mixes less with the aqueous phase, leading to a more efficient extraction. Ethanol precipitation. Nucleic acids can be precipitated from dilute solutions with ethanol. This precipitation can be a concentration step or a means to change buffers, especially after phenol extraction. Typically, either sodium acetate or potassium acetate, pH 5 to 5.5, is added to a final concentration of approximately 0.3 m. The sodium and acidic pH will neutralize the highly charged phosphate backbone and to promote hydrophobic interactions. 2 to 2.5 volumes of 2 to 2.5 volumes of ethanol are added, and the sample is incubated at minus 20 degrees centigrade. If the nucleic acids are small in size and or low in concentration, an extended incubation, several hours, is required. 2. Overnight is needed. The precipitated DNA is collected by centrifugation. The pellet is rinsed with 70% ethanol to remove any excess salts, dried and dissolved in the appropriate buffer. A variation is to substitute ammonium acetate if the hard salts are a problem. Another modification is to use an equal volume of isopropanol instead of 2 to 242 volumes of ethanol, which minimizes the increase in sample volume. Isolation of high molecular weight genomic DNA. High molecular weight chromosomal DNA is usually isolated by multiple rounds of phenol extraction and enzyme treatments, as discussed above. Shear forces, which can break long DNA molecules, need to be avoided during all steps, and sample should never be vortexed. Therefore, the phenol extraction is carried with gentle rocking for several hours. These precautions, again, shear forces are not necessary in the isolation of loss to molecular weight DNA. Another common modification at the ethanol precipitation step is spool out the high molecular weight genomic DNA on the end of a sealed pasture pipette. The precipitated DNA is wrapped around the end of the pipette, is then allowed to partially dry, and then dissolve in the appropriate buffer. This minimizes the contaminants with RNA and low molecular weight DNA fragments. Plasmid mini preps and adsorption methods. Historically, phenol extraction are used for the isolation of most forms of nucleic acids. It is now more common to use techniques based upon adsorption chromatography for the isolation of smaller DNA molecules such as plasmids. Various kits are available for the rapid isolation of small amounts of plasmid DNA. The procedure consists of solubilizing the bacteria in an alkali solution, followed by naturalization with the sodium acetate. The neutralization results in the precipitation of some of the protein and the genomic DNA, which is removed by centrifugation. The soluble material is then mixed with a resin in the presence of chaotropic agents, usually guanidine hydrochloride. The resins are usually either based on silica or diatomaceous earth. Under these conditions, DNA binds to the matrix, but proteins and RNA do not. The DNA is eluted in a low-salt buffer. These methods are rapid and yield a highly purified plasmid DNA, which can generally be used directly in most applications without further processing. Another common application for an adsorption method is the isolation of DNA fragments following gel electrophoresis. In this case, the agarose gel piece containing the DNA is dissolved in NaI, a chaotropic salt, and the DNA adsorbed to silica. The DNA is then eluted with a low salt buffer and sometimes gentle heating. Isolation of RNA. Most RNA isolation protocols also involve phenol extraction and are similar to DNA isolations. However, there are some differences and special considerations. In particular, precautions against RNase activity must be taken. RNase is an extremely stable and active enzyme. Gloves should be worn at all times, and sterile plasticware should be used whenever possible to avoid introducing exogenous RNase to the sample. Glassware needs to be treated with DEPC water and autoclave to inactivate any RNase. Buffers should be prepared from DEPC water or RNase inhibitors included. The cell lysis and solubilization of RNA will typically be carried out in guanidine salts, especially guanidine thiocyanate. Guanidine is a strong chemotropic agent and will inhibit RNases. The strongly denaturing effect of this salt will also promote beta-phenol extraction. The phenol extraction is the same as the DNA isolation, except that the phenol is usually saturated with a buffer of pH 5 to 6. The lower pH will result in some DNA partitioning in the organic phase if DNA contamination is a problem. It is possible to purchase RNase-free DNase. Long-term storage is best as a precipitate in 70% ethanol at minus 20 degrees centigrade. Precipitation of RNA and LICA, that's LICA. So LIL has been used selectively to selectively precipitate RNA. Large RNAs, rRNA, mRNA, are insoluble at high ionic strength, where small RNAs, tRNA, and 5S RNA are RNA. And DNA generally remains soluble. Following either phenol or guanidine extraction, an equal volume of 8 LICL is added. The sample is mixed vigorously and incubated at minus 20 degrees centigrade. The precipitate is collected by centrifugation and reprecipitated if necessary. Affinity chromatography. Most eukaryotic mRNA contains a stretch of A residues at its 3' end, which added post transcriptionally. It is possible to isolate mRNA by affinity chromatography on oligo-T columns. The RNA solution is passed over an oligo-DT column under conditions which promote base pairing. Only RNA with a poly-A tail binds. The poly-A RNA is eluted under conditions, usually lower salt and higher temperatures, which break the base pairing. Density gradient centrifugation. Density gradient centrifugation can also be used in analysis and isolation of nucleic acids. Double-stranded DNA, single-stranded DNA, RNA, and proteins all have different densities and therefore can be separated by isopycnic, that is equilibrium centrifugation. CSIR is the standard medium for density gradient centrifugation of nucleic acids and are especially useful for the purification of large amounts of highly purified DNA. The gradients are carried out in the presence of ethidium bromide, which fluoresces when bound to DNA. The DNA bands are detected by illumination with the UV light, ultraviolet light, and easily recovered with a syringe and needle by puncturing the wall of the disposable tube and aspirating the DNA. The CSCL can be removed by dialysis or by precipitating the DNA. The percentage of guanidine chloride content affects the density of DNA as shown in the figure. This can result in multiple bands on CSCL gradients if DNA compose different G and C compositions are centrifuged. For example, minor bands called satellite DNA are often observed when total DNA from an organism is analyzed by CSCL gradients. These satellite bands are also usually used to highly repetitive DNA or organelle DNA. Mitochondrial DNA is noted for its high 80 strength. These satellite DNA bands can be purified from the genomic DNA by density gradient centrifugation. Nucleic acids can also be separated according to size by rate zonal centrifugation on sucrose gradients. However, this method is not widely used since gel electrophoresis is generally a more convenient method for the size fractionation of nucleic acids. Analysis and quantification. The quality and quantity of isolated nucleic acids can be determined spectrophotometrically. Nucleic acids have an Amax of 260 nanometers and proteins have an Amax of 280 nanometers. The A260/A280 ratio is therefore indicator of the degree of purity of the nucleic acid. A260/A280 ratios of 1.6 to 1.8 or 1.8 to 2 are usually acceptable for DNA and RNA, respectively. The standard extinction coefficient used for ssDNA, dsDNA, and RNA are 0.03 milliliters per microgram or 0.02 milliliters per microgram and and 0.025 milliliters per microgram, respectively. Formulas which take into account protein and other contaminants are also available. Indirect spectrophotometric assays for DNA quantification are also available, but rarely used. It's in some instances, fluorometry using fluorescent dyes that bind DNA and/or RNA is used to determine nucleic acid concentration. Protein isolation and purification. In order to purify protein, you need a source. It might be blood or some other biological fluid, but most of it is a cell, usually a specific type: liver, muscle, yeast, bacteria, etc. And the cells must be broken open, homogenized, to release the protein in a soluble form. Homogenization conditions must be worked out that release the protein from the cell without damaging the protein. Membrane-bound proteins can also be purified, but different approaches are required. Fractional precipitation in concentrated salt solutions, usually ammonium sulfate, is used. Some proteins are more soluble than others. By varying the concentration of ammonium sulfate, one can achieve some limited purification of proteins. This technique is often used in the first step of protein purification. Small proteins are more soluble than large proteins. The larger the number of charge side chains, the more soluble the protein. Column of chromatography. The invention of column chromatography was a critical event in biochemistry because it was the basis for the development of procedures for obtaining pure proteins. Studies of pure proteins are essential for understanding the structural and functional properties of proteins. In column chromatography, an adsorbent is placed in a glass tube, column containing stationary phase. Watch the diagram. The load sample, the next add solvent, and collect the components. Process of column chromatography. A protein mixture is passed into the column and binds to the adsorbent, so on the surface. By proper choice of the eluting buffer, specific proteins can be eluted from the adsorbent and separated from other proteins in the mixture. By repeating this procedure with several different adsorbents, pure protein can be obtained. Because proteins are not very stable, low temperature 4 degrees centigrade and neutral pH must often be employed. The properties of some adsorbents are described below. Ion exchange chromatography. Ion exchange resins have fixed charges, either positive or negative. Proteins bind to the resin by electrostatic interactions. The strength of these interactions depends on the net charge of the protein, which is a function of pH, and the nature of the weak amino acid side chains and the salt concentration. The buffer high salt concentration reduces the interaction. Affinity chromatography. This is a more specific interaction in which a ligand specifically recognized by the protein of interest is attached to the column material. When a mixture of protein is passed through the column, only those few that bind strongly to the ligand will stick, while others will pass through the column. By changing the buffer, one weakens the interaction between the protein and the ligand, which causes the protein to be eluted from the column. A variation in immunoaffinity chromatography, in which an antibody specific for a protein is immobilized on the column and used to affinity purify the specific protein. This is a diagram of a typical curve of protein during chromatography, and the topic will be gel filtration chromatography, molecular sieve, or size exclusion chromatography. Before that, have a look at the diagram. There's a relative amount going upwards and on the vertical axis, and the other one elution volume, ml, on the horizontal axis. Now the column consists of material that separates proteins based on their size and shape. A wide range of molecular exclusion limits is available to separating proteins of all sizes. For any particular column dimension and the material, the volume of buffer required is to elute a specific protein depends on the molecular weight of the protein, thus one can separate proteins by size. If one calibrates the column by determining the elution volume of proteins with known molecular weights, then a calibration curve relating elution volume and molecular weight can be constructed. Such a calibration curve can then be used to estimate the molecular weight of an unknown protein. Dialysis or ultrafiltration. Semi-permeable membranes are available, which allow passage of small molecules but exclude the passage of proteins. Sacks made of such material allow the salt and the buffer components of a protein solution to be changed to another buffer. Continuing with the last portion, dialysis and ultrafiltration. Semi-permeable membranes are available which allow passage of small molecules but exclude the passage of proteins. Sacks made of such material allow the salt and buffer components of a protein solution to be changed to another buffer. And the analysis of DNA, RNA, and protein. DNA and RNA form the nucleic acid, while protein is the structural macromolecule of the cell. These are analyzed by electrophoresis, centrifugation, etc. Now, we go to look at the diagram. Shows salt, protein, and salt. There's a dialysis bagger from which salt enters, salt leaves as well. Large volume of buffer solution with salt. And this is a process of dialysis. Now we go to the next topic of gel electrophoresis. Electrophoresis of macromolecules can be carried out in solution. However, the ability to separate molecules is compromised by the diffusion. Greater resolution is achieved if electrophoresis is carried out on semi-solid supports, such as polyacrylamide or agarose gels. Gels are formed by cross-linking polymers in aqueous solution medium. This will form a three-dimensional meshwork with which the molecules must pass through. Polyacrylamide is a common gel for protein electrophoresis, whereas agarose is more commonly used for nucleic acids. Agarose gels have a larger pore size than acrylamide gels and are better suited for larger macromolecules. However, either type of gel can be applied to either nucleic acids or proteins, depending on the applications. Gels are formed from long polymers in a cross-linked lattice. The spaces between the polymers are the pores. Higher concentration of the polymer will result in smaller average pore sizes. Polyacrylamide gels are formed by covalently cross-linking acrylamide monomers with bis-acrylamide with a free radical like persulfate SO4. The cross-linking of the acrylamide polymers results in pores of a defined size. The total acrylamide concentration and the ratio of bis-acrylamide to acrylamide will determine the average pore size. The polyacrylamide solution is poured into a mold and polymerized. This mold can be a cylindrical tube, but aging is usually a slab poured between two glass plates. Since the gel is solid, with respect to the mold, and molecules are forced through the gel, smaller molecules will be able to pass through the lattice more easily, resulting in larger molecules having a lower mobility than smaller molecules. In other words, the gel acts like a molecular sieve and retains the larger molecules while letting the smaller ones pass through. This is the opposite of gel filtration, where the larger molecules have a higher mobility because they do not enter the gel. Therefore, the frictional coefficient is relative to how easily a protein passes through the pores of the gel, and size will be the major determinant of the mobility of molecules in the gel matrix. Protein shape and other factors will still affect mobility, but to a lesser extent. Substituting size for the frictional coefficient results in mobility, which is equal to voltage, charge by size. Equipment. Equipment to conduct gel electrophoresis is relatively simple. They consist of a mold to form the gels, an apparatus to hold the gel and contain buffers, and a power supply capable of delivering the required voltage or charge current. There are many types of apparatus for carrying out electrophoresis depending on the application. Gels can be either in a vertical or horizontal configuration. Polyacrylamide gels are run in a vertical fashion, and agarose gels tend to be run in a horizontal position. Gels can either be formed as cylinders by using glass tubing as a mold, often called tube gels, or formed as a rectangular slab. These slab gels are formed by polymerizing the acrylamide solution between glass plates separated by spaces. Typically, the gel is 0.75 to 1.5 millimeter thick. At the top, a comb is used to form sample wells. Slab gels allow multiple samples to be compared on the same gel, thus eliminating gel-to-gel variations. The formed gel is placed into the apparatus to that the top and the bottom of the gel are in contact, so that they are in contact with chambers containing buffer. These chambers contain electrodes which are connected to a power supply. Thus, an electric field is generated across the gel, which, when a voltage is applied, the buffer in the 6 chambers is generally different, but the buffer making up the gel for protein electrophoresis, and in some applications the buffers in the lower and upper chambers may be different. In most applications, the buffers are such that the protein has a negative charge, and therefore the anode positive pole will be in the lower chamber and the cathode negative pole will be in the upper chamber. However, there are applications in which the proteins of interest may be positively charged, and therefore the electrodes will be reversed. This is a picture of the illustration or schematic of the buffer and the gel and a high voltage supply about the process of electrophoresis. Protein characterization. Excuse me. SDS page sodium dodecyl sulfate polyacrylamide gel electrophoresis is a variant of electrophoresis in which the buffers contain SDS, a detergent that binds to proteins. I think you might be remembering the previous lecture I was giving you about the latest reviews. I was talking about this, right? So keep referring to those notes as well. Most proteins bind SDS at a constant ratio, about 1 SDS for every 2 amino acids. The large negative charge resulting from the bound SDS masks the negative charge on the protein, so that all proteins have essentially the same charge to mass ratio. This means that the rate of movement in the electric field depends only on the polypeptide chain molecular weight. Mercaptoethanol is used to reduce disulfides. In addition, the SDS causes all proteins to adopt a random coil structure, which means that shape does not affect movement through the gel. The SDS page is a very useful method for determining the molecular weight of the individual polypeptide chains of a protein. Western blotting is a technique for detecting a specific protein in a mixture. Gel electrophoresis and SDS page electrophoresis are primarily useful as analytical techniques, although they can be used for purification. Now, this is a typical SDS page gel. In this figure, lane 1 would contain standards of known molecular weight. Lane 2, a mixture of 3 unknown proteins, and lane 3 to 5, 3 unknown proteins. SDS page can be used to determine the molecular weights of a protein. The molecular weights of the 3 unknown proteins can be determined from a calibration curve constructed by plotting the log of the molecular weight of the standard proteins versus the distance traveled in the gel. The distance traveled depends on the porosity of the gel. The lower the porosity, the faster further the proteins move. Depending on the molecular aspect of the unknown proteins, different porosity gels will need to be used. Isoelectric focusing. Isoelectric focusing, IEF, separates proteins based on their isoelectric points. The isoelectric point is defined as the pH at which a protein has no net charge, that is, the number of negative and positive charges are equal, and is a measure of the protein's net charge. Separating proteins according to their net charge is accomplished by generating a pH gradient in an electric field. The effect of protein size on mobility is minimized by carrying out the electrophoresis gels with large pore sizes such as low acrylamide concentrations. Example 3.5% of agarose. This large pore size minimizes the molecular sieving. A pH gradient is generated with carrier amphilites. These amphilites are a mixture of aliphatic amines and either carboxylic or sulfonic acid. These ampholytes are a mixture of aliphatic amines and either carboxylic or sulfonic acid. They have a high buffering capacity, low molecular weight, 300 to 600 Da, that will be Dalton's gas, and a range of pKa values. Initially, the pH of an ampholyte solution will be the average of the pK values. The mixture application of an electric current will cause the ampholytes to migrate towards the electrodes according to their charges. Ampholytes that have pKa values above the pH will be positively charged, and those with pKa values below the pH will be negatively charged. As the ampholytes migrate, this will result in changes in the local pH due to the buffering action of the ampholytes. This change in the local pH will affect the charge of the ampholytes. Depending upon the pKa, the ampholytes will continue to migrate until they reach a position in which the local pH equals their pKa, that is not a net charge. The end result is a pH gradient in which the most basic ampholytes are found at the cathode. A dilute alkali solution, example sodium hydroxide, and the most acidic ampholytes are at the anode. A dilute acid solution, example H1PO, okay. Carrier ampholytes with defined pH ranges can be purchased or prepared by isoelectric focusing. Proteins are also ampholytes and will migrate with the pH gradient until they reach a pH equal to this isoelectric point. The carrier ampholytes are needed since the protein concentration is generally not high enough to establish a stable pH gradient, and the isoelectric points of the proteins may not be uniformly distributed along a pH gradient. Sample preparation is important for IEF in that many reagents can adversely affect isoelectric focusing. In particular, the ionic strength should be as low as possible. Precipitation of proteins with acetone is a method of removal of excess salts as well as concentrating the proteins. Separation can be performed under either native or denaturing conditions. Urea is the preferred denaturing agent since it is uncharged. Similarly, non-ionic detergents such as Triton X100 or MP40 are less likely to interfere with the formation of pH gradients. Protein precipitation is sometimes a problem in that proteins tend to be less soluble at their isoelectric points and their local concentrations can be quite high. In addition, the high voltages and high resistance associated with IEF generate substantial heat, which increases protein precipitation. Many apparatuses for IEF will have a cooling mechanism to disperse the excess heat. Inclusion of urea and NP40 in the gels will also minimize protein precipitation. IEF is an equilibrium phenomenon since the components of system migrate until they have no net charge. As the system approaches the equilibrium, the resistance approaches infinity since there are no ions to conduct the current. However, the pH gradient will start to break down before true equilibrium is reached, and the ampholytes will migrate into the anode and the cathode buffers. This gradient breakdown is accompanied by a lowering of the resistance. And therefore, the progress of IEF can be followed by performing the electrophoresis under constant voltage and monitoring the current. Initially, the current would rapidly drop in accordance with the rapid migration of ampholytes. As the ampholytes lose their net charge, the resistance increases and the current decreases E is equal to IR. The rate at which the current decreases levels off as the system approaches equilibrium. The current will start to rise again when the pH gradient starts to break down. IEF needs to be discontinued before this point. The pH gradient can be determined with marker pore proteins with known isoelectric points or by measuring the pH along with the gel. This is accomplished by slicing the gel into pieces, loading the ampholyte into distilled water and measuring the pH. Two-dimensional gel electrophoresis. Conventional electrophoresis separates proteins according to their charge by mass ratio. STS page separates proteins according to subunit or polypeptide mass. IEF separates proteins according to isoelectric protein point or charge. It is possible to sequentially combine the different types of electrophoresis and run two-dimensional 2D gels. A common form in 2D gel electrophoresis is to first separate proteins by IEF in tube gels. These is also then equilibrated in SDS gel electrophoresis sample buffer and subjected to SDS page in a slab gel. It is necessary to carry out the IEF first sense SDS interfere with the isoelectric focusing. The 2D separation results in higher resolution since proteins are being separated according to two distinctive properties, that is charge and size. This is a diagram of the SDS page and the IEF IPG, two-dimensional electrophoresis. Molecular weight and shape are fundamental physical properties of a protein. Estimates of molecular weight can be obtained using SDS page or gel filtration as described above. One very useful technique for measuring molecular weight and shape is centrifugation. Centrifugation, a particle that is subjected to a centrifugal field by being spun in a centrifuge is subjected to a force F is equal to M into 1 minus Vp by omega squared R. I think that's omega, where M is the mass of the particle, R is the distance of the particle from the center of rotation, and W is the angular velocity. 1 minus nr is the buoyancy factor, which account for the fact that particle is buoyed up by surrounding solvent density, R gram per milliliter. V is the specific volume of the particle. V is the specific volume of the particle. ml per g is equal to 1 by density of the particle. If y is equal to p, then the particle will not move. The movement of the particle through the solvent is resisted by a friction coefficient F that depends on the shape of the particle.